Welcome to Diagnosing Dx, the podcast giving diagnostics the platform it deserves. While therapeutics and devices often take centre stage, diagnostics is the engine driving precision medicine. I'm your host, David Philby, and each week we spotlight the innovators, breakthroughs, and real-world challenges shaping the future of DX. This week on Diagnosing DX, I sat down with Vincent Funari, Chief Scientific Officer and Genomics leader whose journey spans the Human Genome Project to liquid biopsy innovation. We talked about scaling NeoGenomics, leading during COVID, and landing FDA praise for groundbreaking assays. Vincent also shared what's next in cancer diagnostics and how he went from bench scientist to Chief Scientific Officer. Let's dive in. Well, welcome, Vincent, to the show. Thank you so much for coming on.
Yeah, it's good to be here. Really excited to talk with you. So,
thank you so much. I've been really looking forward to it because I think when we caught up and we sort of naturally started unpicking the industry, the landscape, and I guess a lot of change that's going on. So I think we've got a lot about the past to talk about, a lot about the present, and a lot about the future. I guess for the purposes for the benefit of the audience, it'd be great if we kick off with a bit of an intro into your background, who you are, and what you've been doing.
Yeah, that'd be great. I often tell the story. You know, it kind of dates me, but I started back when there wasn't a human genome. For those of you that can remember that, that's where I started with the Human Genome Project. I was lucky enough to be in a lab, genetics lab, and I have a number of bioinformatic advisors around me to start mining the data. And so I was one of the first to graduate with a thesis in bioinformatics. And so that was pretty exciting. And then I moved on, hoping that genomics was going to be the next revolution. Yeah. And so I moved on to Stanford and learned all about gene expression profiling, which was really exciting. I was focusing on breast cancer there. And then moved to Cedars, Sinai, and UCLA. And that was probably one of my most memorable experiences, spending about 10 years in academia and opening up my own lab for genomics and being able to work with a lot of amazing people doing amazing things. And I was lucky enough to work with them and support them with genomics research. And then after that, I realized that I really wanted to see this move into the clinical domain, genomics in the clinical domain, and it struggled with microarrays, but I saw next-gen sequencing as an opportunity to do that. So I moved into NeoGenomics, which was a smaller company at that time, probably about 50 million in revenue. They were based in Irvine. And so I moved out to there and had an amazing advisor that supported genomics into the clinic. And so we built servers, bioinformatic pipelines, and assays to support oncology testing diagnostics. So I spent a good 7 years there. We developed a lot of assays, developed the infrastructure, supported pharmacology, and that was a very exciting time in the industry, as you probably know. A lot of growth everywhere. And so after that, I had had a lot of fun and wanted to start something fresh and new, and different. And that was essentially moving my job outside of the US, actually experimenting with decentralized diagnostics in industry and in Canada. So that was pretty exciting, going international. I learned a lot about the different regulatory processes, and then moved on to Emeritus Dx, which is a smaller company again, and focused on cancer and infectious disease. And so spread my wings a little bit, doing more other things besides genomics, getting back to digital PCR and other things.
Wow. A lot to unpack, Vincent. So you mentioned when you joined NeoGenomics that actually they were a relatively small company, and by the time you left as a chief scientific officer over 7 years, that company would have been through an incredible journey. So when you first joined NeoGenomics, what was it that drew you to them? And as scientific expert and background, was there something that you felt they were the best company at that time that was going to sort of scratch the itch on what you were most passionate about?
Yeah, that's a really good question. You think a lot about, when you move someplace that, especially when you're comfortable. I was very, very comfortable and loved my job at Cedars. So it was the opportunity. I saw that the chief medical officer there really was supporting and really engaged in next generation sequencing and genomics. And that excited me a lot. I had started a precision medicine committee at Cedars, along with a number of really good scientists and physicians. And we evaluated it at Cedars, but I saw industry moving just a lot faster.
Right.
And so looking at Neo, it was a smaller company, but the vision I thought was there, and the people that could drive the vision were there as well. And so I think that excited me, if anything, and taught me there may be an opportunity there to grow with them.
It's amazing how many times we hear that the people that you have the opportunity to surround yourself with and be on the journey with can, if there's something scientifically that's going to get you out of bed in the morning, even that can be sort of overruled by the team, the people that are there and the culture they're trying to harness and create, and therefore the ability to innovate and, to your point, to move at a certain pace in innovation can be really exciting. So what did that team look like when you joined? How big was Neo, and sort of, what were they doing at the time?
Yeah, it was small. I'm not gonna lie, it was small. So there was an opportunity, but there was also a risk. But but again, like exactly what you said with the team and everything. But it looked like we didn't have any bioinformaticists, so it was just me. So I had to help build the cluster and servers and infrastructure, pipelines, and everything like that. And as far as assay development, they had a really good start. I understood what they were trying to do. Um, and the chief medical officer, again, it was small. So we were utilizing, it was sort of multitasking, if you will. So there were people in operations that supported us a lot, where in a lot of companies, a lot of the development is done solely in R&D. But when we first started, we didn't have a lot of people, in R&D. And so we need to depend on others that would participate in the R&D functions. And a lot of that came from operations and other people, whether it be IT or whoever.
And so how did your role develop from the role you were brought in to do initially to over the 7 years?
Yeah, yeah. So as the company grew, we started to do more acquisitions. In fact, in the first 6 months, we acquired a company that was twice the size of our company. So we were very profitable to start with, which was good. And we had an amazing CEO that really, again, supported and helped foster this. And so my role started to change really when those deals started to occur. I think the M&A deals, I had to support the new infrastructure, bringing people into my group, which was a new challenge for me. Usually you go out and you hire your own people, but when you're doing these M&A deals, you're acquiring a company and everything that comes with it, including culture, infrastructure, and trying to bring those people into what Neo's culture was and what our infrastructure was always a challenge for all the deals we did, which really, again, there were a lot of deals over the 7 years that really grew the company to where it is today. And on top of that, as the company was growing, there were a number of changes in, I would say, the executive group or people that were mentoring, everybody in the company. And so when those changes happened, there were opportunities. And I think I was in a place where I was able to gain the trust and work with different executives, et cetera, the CEO, and to help to help with this growth. And so it was really an opportunistic as well as, just the opportunities were there.
Yeah. When you're in them acquisitions, are you finding that the teams that you're bringing into the company, do they tend to have excitement to now be part of NeoGenomics, or do they are they potentially disgruntled and not excited about the acquisition, or is it case dependent on what's happened and why it's happened?
Yeah, that's a great point. I think in general, they were excited to be folded into a company that was profitable, that was growing, and was stable. Many of these M&As that we did, the acquisitions, I think were done with companies that were small and trying to grow, but there was still an unstableness feeling in.
That makes sense, because we see this all the time in the industry at the moment, right? In terms of small companies that have some really exciting science and a great team, but are just running out of cash runway. This is probably going to be a big year for acquisitions of that side, and the stability, in a word, is ultimately scientifically what you want, isn't it? The ability to keep doing your research when you feel like it's going to go somewhere and it's going to have a big impact. Do you feel that this is, I don't know how you're going to answer this, but do you feel that R&D teams, bioinformatic teams are an easy group of people to integrate into new environments and new cultures? At Alevra Consulting, we focus on DX only, supporting early stage and growth companies achieve their milestones by solving their biggest talent challenges. Have you ever suffered from working with a talent partner who was either life sciences or med tech and dabbling in diagnostics? Or no more? Work with a diagnostics specialist talent partner.
So that was a new experience for me. I'm going to say that I think it was a challenge. The cultures are oftentimes a little bit different.
Yeah.
So what they've grown into, especially the younger scientists that are probably around, what they've been used to, what they've been told, and then in an acquisition, it's a little bit of a surprise for them. So I think it's a little bit of a challenge to grow and mentor through acquisitions. The big joke was integrations. We would say it would take a year, and of course it would take 2 years or 3 years to fully integrate. Find out where people best belong, because it might be different. Their R&D department and bioinformatics group might be different than ours. They may have different specialties. They may be focused on different things. So maybe they're better at pharmacogenomics, and although they call themselves a clinical division, maybe better at pharmacogenomics. So trying to find the best fit for them is important for the company and for them and for us, for everybody, for our culture. So yeah, it's a little bit of a struggle. Yeah.
Yeah, super interesting. These acquisitions, they always tend to take much longer than than is expected. And actually, a lot of them end up just operating as sort of siloed teams under the same brand, right? And the integration is actually more of a operational. Yeah, they're now part of the same company, and we've been able to remove a bunch of cost because we already have these departments here, especially back office and support and stuff. But ultimately, the work they're doing just continues in a siloed nature. And so, what? So over the course of there, so in terms of the milestones that you were achieving and that NeoGenomics were hitting over the 7 years there, because it's been a successful period. Give me an overview of the sort of things that happened in your time there.
Of course. So one of the really great things, again, when I was there, was the support for a new development. And so some of the milestones that we had were developing one of the first clinical super sensitive assays that included novel bioinformatics and novel assay development. And I think that was very exciting. On top of that, because some of the other milestones I think about that were pretty exciting, where it was a novel gene fusion and expression assay for both heme and solid tumor. And those really were at the forefront, I would say, of the industry. People really didn't focus on gene fusions and our expression like we did. We realized the importance of it. And so that was very exciting. I would say, in addition to that, we developed one of the first heme assays that we were working with the FDA to get approval. It was a de novo assay. And so working with the FDA on a de novo assay, I feel was one of the highlights. I got to really drive that effort. And that was really exciting to work with the FDA on that. And at the end, the FDA called, and to that point, the FDA called our application the best heme application that they had seen.
Wow.
So that was really exciting. And then same thing with Medicare. Medicare thought our gene fusion assay was the best one they had seen. They actually had no comments when we submitted to them, which is really rare if you know anything about submitting to Medicare or New York State, which are really important in the regulatory space for a centralized lab like ours. That you're going to get paid either by the insurance or you're going to get paid by Medicare, and so being able to get those paid was really important.
Super interesting. So you'd been with NeoGenomics 7 years. It was your first major step into the industry side of it outside of academics, and you took that leap. You said there was risk, right? What was the greatest lessons learned and the greatest memories of that time? Now you look back on that and having made that move.
Yeah, so I think one of the memories that I have with NeoGenomics was this thing came along called COVID, and it really disrupted not only oncology testing, because as you know, there were probably many people that didn't really go in to their docs at that time. They didn't want to go into the hospitals, etc. So it really changed our main focus, our main business. But at the same time, also, there was a lack of testing done during COVID. And that was a big issue, especially in California. And there were many hurdles to do that testing, bringing it up an infectious disease department, essentially. And in addition, trying to get supplies. Everybody was out of supplies, so trying to make sure you had supplies, and just the upscale. So at Neo, maybe we did 1,000 tests a day, and now we were being asked to do as many as 50,000 tests a day. That was actually the goal we had to support the infrastructure for the country and for California. And so that was a very memorable time because it really required a concerted effort among all the different departments and divisions, whether it was IT, whether it was reimbursement, whatever it was, it required a real integrated effort. And so that was a really memorable moment, I would say, when I was at Neo. It was probably when we talked to people, it was one of the most exciting moments, pushing the envelopes of assay development and scaling and doing things that we had never imagined that we would be asked to do. So I think that was a very memorable time, and so glad we're in a different world today. But that was an example of real team effort.
Yeah. You must have felt like you were on the front line of COVID at times, right? If you're being asked to be responsible for so much testing, because I mean, even as you know, I was living in New York City at the time, and I felt like I was living in the epicenter of COVID at times, and it was like a zombie town, right? But I could only imagine, that feeling when it was so uncertain, right? The world, and we just didn't know what was coming next, and for you to be involved in a company that was at the forefront of testing, diagnosis, and so forth, it must have been such a rare feeling to be at the forefront of something like that.
Yeah, I think that's a great observation. It was a time that you were talking to your friends, your neighbors, whoever it was, and everybody was in the same place where everybody was
Yeah,
scared to go out, like you
yeah,
said, at the ghost town in New York. That was everywhere. Everything stopped. I have little kids and they were in sports and what have you. That changed and that stopped.
mm,
So it was really felt good, I would say, to be able to try and be, like you said, at the epicenter of, some of the testing and to try and really support the country through the times. And that included everybody that I would meet, our friends,
yeah.
our neighbors, whoever it was. So that was good. Yeah, I agree. I agree.
So let's move on to current day and where you are. So I mean, you're still at the front line in many ways of where the industry is going in terms of innovation and what's coming next. What do you feel is a bit? I mean, this is really why I wanted to have you on. Really, is to sort of understand from your perspective and what you're seeing is what's really exciting in the space at the moment, and in terms of innovation and innovation in the industry, and what do we think is coming next, and what are you excited about?
So that's a great question. And so I think it's important for people to understand what's going on today and how that differs from where we envision it going, which is, I think, a very different mindset. We started down the path of genetics, right, for cancer and trying to understand that. And so we developed a number of genomic testing to do that, And so what's next? Well, the sensitivity of these assays, I think, is on the verge of being able to support things like, early cancer detection, where you might go in and get a blood draw, and while they're testing for your potassium levels and your PSA levels or whatever it is, while they're doing that, they're looking for cancer, and so that's a really exciting day when we get to there. I think we're a little ways off, but that's exciting. We're currently in a little bit of a revolution right now in cancer oncology or cancer medicine, and that is people are turning to liquid biopsies in preference over the gold standard. The gold standard is solid tumor testing
Yeah,
or blood or bone marrow testing, and people are turning to liquid biopsy. And the rationale, so as one of the key leaders of our scientific advisory board at Neil, I'll never forget. We had one of the key opinion leaders in lung cancer, and when we were talking about solid tumor testing and the gold standard, he at that time was saying that he would prefer, and this was again like 5 years ago or so, but he would prefer liquid biopsy, and the rationale was that you can get as much blood as you need and be able to send it off and go
Yeah.
back and get more blood and what have
Yeah.
you. But one of the key things was when the patient comes in, they can do a blood draw, and even before anything, they could send it off for testing. Before the MRI, before the surgery, before all of that, such that when they do the surgery and do the traditional anatomical pathology stains and those things, when they do that, they all already know here are the therapeutic options that are available, what the prognostic, prognosticating the tumor for the patient and trying to come up with a treatment plan. But they could do that really early versus a lot of times if you wait to get that surgery, wait to get that biopsy, and then send it in, and then have all this testing done. Genomic testing takes a while. It's not something that you can do overnight. It's not one of those tests. It's something that takes a while. So to be able to do that at the forefront, I think is really important and is really coming of value. So one of the other exciting new, I think, technologies to arrive in cancer diagnostics is minimal residual disease testing. And so that's a really exciting opportunity because being able to monitor the patient over time and to identify whether or not the cancer is coming back or the treatment is working before you can go do your MRIs and CAT. So before something shows up on an image, you're getting information already, and you're getting genetic information which may help you change your treatment depending on what you get. There are resistance mutations that are come up as you're being treated to certain therapies, and so to know when that's happening so you can change your treatment, I think is really important and a new field, exciting field is coming up. The challenging part with all of this is putting it into operations.
Right.
So it's great we can do this with research, but then, when you're a doctor and you're seeing some of this, how do you interpret it? You don't have all that testing, docs, in my experience, that are doing oncologists that are doing the therapy selection and trying to monitor the patient. They are relying on traditional, information that it's been vetted many times around by other oncologists, specialists in the field, etc., such that they become part of the cancer testing guidelines. And so a lot of the stuff is ahead of the curve. And so it's not in the guidelines yet,
Yeah.
but it will be at one point. But how do you get from not being in the guidelines to being in the guidelines is a bit of time, even though you may have a great assay. It may be perfect for monitoring the patient, helping your patient get better. It's still a bit of a risk in the oncologist's minds. So they're very careful about treating their patients.
Yeah.
But, which makes, that's what we want.
Is it that it's is it seen as a risk, or is it that doctors can be stubborn and they've got if they've got the gold standard, if they're used to sort of tissue based biopsy and that's the way they've done it for their career, depending on their age, how long they've been doing it, and then suddenly, liquid biopsy comes along. I know we're at the point now of, FDA approvals and Guardant Health have brought, a lot to the market. I actually heard recently that I think there's somewhere between sort of 60 to 70 liquid biopsy early cancer detection tests in development as at any one time in the US or something like that. It's a significant number. But uptake is is not just, proving, a lack of risk and like you say, getting it into guidelines, but it's also changing physicians' opinion and doctors' opinions on whether they need it, right? And whether it's going to improve them. Do they want to change their processes?
Yeah, I think that goes along with any part of medicine, right?
Right.
Like, so you may go in and your friend told you about this one test, then another physician did or uses, and you ask about it. And depending on the physician's comfort level, working with the patient, etc., it, to your point, it depends on the physician who is maybe more likely to adopt one of these newer methods and change practice. It's challenging because they then not only have to interpret it, but they have to explain it too. And explaining genetics to the common population, as you can imagine,
Yeah.
is a challenge. So it's them knowing the field, it's them being comfortable with trying something that's cutting edge, and then them being able to take that information and put it in a way that the patient can understand what's going on. So, yeah, I think it's very physician dependent on who adopts what, and it's their comfort level, like you said. Some physicians are really comfortable with genetics, genomics, and being able to know what's going on. Maybe those are the younger generation. Maybe the older ones are more comfortable with what has worked in the past and
Right.
they know works because they've had the experience, right? So the older ones are ones that have tried what was in the guidelines and they know
Yeah.
it works. And then so they've seen it work in their hands. And so do they really risk going outside of that?
Yeah. And I think there's a bit of a trend in the industry right now that there's a lot of early stage companies that, don't have FDA approvals, but have their clear lab set up, maybe a cap lab, maybe New York State, but let's even just companies with clear labs operating through their lab developed test model. And they're being, at the moment in this landscape, they're being forced to drive revenue, commercialize, because ultimately capital is so tight and the ability to raise is so tight. Investors want more proof of market. That feels like an uphill battle for a lot of companies that are really sort of pushing into commercialization and sort of really trying to dial that up at the moment. But that that feels that's very common at the moment. I speak to a lot of companies that are early stage and having to really ramp up commercial activity through their LDT setup at the moment.
So, David, in your experience, do you think that's becoming more of a challenge in these days versus, 10, 15 years ago? Or do you think the challenge has always been there and just the field may change, but the challenge of getting investment for, new development and new tests is harder now than it was before?
I think it's I think there's a couple of macro situations that we're playing with. Interest rates have been high for 2, 3 years now on the back of high inflation and COVID. Interest rates in the US, the Fed has acted very slowly. We still haven't seen that come down, which will inevitably create more liquidity in the market. And early innovative industries like diagnostics, med device, med tech, biotech, and even software, excluding AI, because that's the outlier at the moment.
Yeah,
But
right.
innovation based industries, which the US is known for and thrives upon, are struggling from a lack of liquidity. And then I do also think that as we sit here today, we're in the first year of a new administration, and every new administration causes problems in the first year when they try and implement their problems, because if you can get them done by the time the next election run comes around, people have forgotten that, and you hopefully have got results for your actions that you can put
Eh.
out there as your, for want of a better term, propaganda. But I think it's difficult right now to raise capital. I think that's definitely true. But I think the challenge is knee-jerk decisions when early stage companies have raised money previously, have gone back to the well and said, now we need a bit more. And them investors have said, not until you've proven you can make more money. And at that point, they've gone, well, we weren't expecting this. We thought,
Right.
because of our previous conversations, that we were going to be able to come back and get the next, add on or top up to get to where we need to be. But now they're being asked to suddenly emphasize more on revenue generation and profitability now. And that's putting a lot of stress on companies. And that sort of goes back to what we said earlier about acquisition targets, and the bigger firms collecting a few of them early stage development firms in at good prices right now, which is just the way it is. That's just the way the market moves. But yeah, it's an interesting dynamic, that, and I think everyone prays for a bit of relief, right? And I think I'm hoping for sort of better conditions next year. Based on those two macro situations, I think the macro of interest rates being really high, the uncertainty in what's happening globally at the moment, and them two things settling down should hopefully yield a better 2026 for the capital raising market.
Yeah, and I'll throw into that mix. You probably are aware of this, but the changes in the regulatory environment
Yep.
that are actively going on, right? Like,
Yep.
so I think people recognize that there needs to be a little bit more oversight in that department. However, what that looks like, will it stifle, a lot of these small companies that you're talking about?
Yeah.
And the only ones that will survive will be, the very large ones. So, that has yet to play itself out, but I think that's also part, I would put on the third bullet point on your talk would be, the uncertain regulatory,
Yeah.
environment.
Yeah, 100%. That's certainly not been finalized, even though we've got probably a good decision on that for the early stage firms at the moment. They're probably, really excited about that, but
Yeah.
it's certainly not the end of it. So
Right.
let's do this, because I think it's really important we get to ask this question. And scientific background doesn't, well, I think it splits the opinion in terms of people following their passion. That whenever I speak to scientific backgrounds, they either love to stay close to the science and have no ambitions to lead, manage people or
Right.
operations or anything,
Right.
or, as you have done, you ended up as a chief scientific officer. What's your experience of getting to that level? What's enjoyable about it, and what's not?
So, and I'm going to harken back to my days in academia. It was a similar situation where you had you probably have this ingrained in your mind when you start, that you're going to start at one level, then eventually you're going to be a bit more independent, and that turns into having your own ideas and being able to make those come to fruition. And that is something that you get as you grow in these parts. So, when I started off as a scientist, before I had my lab, I loved exactly what you said, just being in the science, not worrying about politics,
Yeah.
not worrying about, things like, the culture in my group. Were people put in the right place, or do I have to move people? Do I have to hire and fire people?
Mm-hmm.
Budget,
Mm-hmm.
assignments. Those are all things that are not science related.
Yeah.
And so you have to be able to learn that and be able to want to do it. Just like you said, and some people just don't want to do that. They want to spend their time in science.
Yeah.
So I was a little different. I wanted to learn those things. I think my mindset going into a PhD was to learn as much as I can. You probably have heard this before, but I like this sort of analogy, which is, when you first start your PhD, you think you know everything.
Yeah.
You really do. And then you start on this one subject, one small little subject in the mass of questions that are out there in the scientific area. And, so one out of a million questions that science has, you start on that and you think it's going to be super easy and you think you're going to know everything. And by the end, you're just like, oh my God, I know nothing, you know? Um, and it's that moment, I think, that you either start to appreciate wanting to do more in the world and try and I think try and be independent, like I said, and trying because it's a real challenge and honestly, at the end, you start your doctorate or whatever training, and you think, oh, I love this. This is going to be great. I'm so
Yeah.
smart. This is awesome. And then you leave and you're a bit downtrodden, a bit frustrated because there's so much more you could do, so much more you can understand, and you've spent years doing this. And so how is this possible? So you end up being a little bit sort of depressed, challenged. I don't know what you want to call it. After that, it's a little bit of a post-traumatic stress disorder. I don't know. It's one of those things.
Yeah.
And so if you feel that you like those challenges and you like learning, and even if you're going to make mistakes along the way, I think you're going to be one of those people that wants to, like you said, I think, do more than just be just science. And I don't mean that in a bad way. It's not
No,
just
not at all.
science. It's doing more than just that one discipline, versus really being at the scientific level. And I can tell you now that I've done both.
Yeah.
And honestly, I think it goes back and forth now that I've done, whatever the highest level that you typically achieve as a scientist. So now that I've done both, I feel that I could literally go back tomorrow and just be a scientist. I'm totally comfortable with that.
That's awesome.
Yeah, yeah. It's a good feeling. It's a good feeling.
Did you early on, did you feel your personality was stood out and different, or was there traits of your personality that was different to the wider team within like an R&D department that you could see others were sort of more they were enjoying different things about the role?
Yeah. So I think early on, when I had started to pursue this discipline, I think I was told by many that I was very much an extrovert, very
Mm,
much a person that can communicate, at all levels, and make people feel comfortable, make uh, talk again, at a scientific level, but also just talk to the the wide range of other disciplines out there,
yeah.
whether it be economics, whether it be strategy, whatever it be. And so I was told that, which made me feel good. A lot of scientists, when you go into being a scientist, I think you know this, David, from talking to people, it's a very introverted
Sure.
field. So I mentored and worked with a lot of people that were exactly as you say, wanting to stay and just do science. And those tended to be introverted people that really just wanted to work and stay within their zone. They didn't want to deal with all the other things that comes with some of the other roles.
Yeah. So then it's about following what you're good at, but also what you're enjoying, and what is passionate for you at that point, what you want to keep doing, and following that path.
Yeah, it is. It's a
Super
mix.
interesting.
It's totally a mix of those things, I think.
That's awesome. But Vincent, I think this has been great, to be honest. I think the focus on your background and learning about the journey in NeoGenomics, the current day outlook on what's exciting in the space, but also your insight and advice on getting to a chief scientific officer level, and why you stood out amongst the pack is super insightful. I've really enjoyed the conversation. It's been great to have you on the show.
Thank you, David. And again, thank you for the invitation. I really enjoyed this conversation. I think it's one of those things that, that I think as a young scientist, for those young scientists that may be listening, I think it's really important to sort of value, who you are, and it's okay to want to do science. It's okay to want to do, multiple roles. It's okay to move into a different field. So a lot of scientists move into sales, or a lot of my colleagues and friends have moved into marketing
Hmm.
at these companies, scientific affairs, is another opportunity. So it doesn't mean that you're sitting at a bench as a scientist. There's so many more opportunities, and it's just understanding who you are, like you said, and making yourself, that they always say, if you enjoy your work, it's not work. You're never working. So whatever that is.
Yes. Good advice. Thank you very much, Vincent. I really appreciate your time. This was great.
Yeah, loved it. Thank you, David.